Hello everyone, I am Harshini along with Anurag, Seema, Swastika, Sagarika, Satvik and Ujjwal would like to present to you about the topic application of horizontal gene transfer in biotechnology. Context. In this, we are going to discuss about the introduction to horizontal gene transfer and application of horizontal gene transfer in biotechnology and challenges and future directions, and at the end let's conclude the topic with a brief summary. Let's begin with the introduction. Horizontal gene transfer, also known as lateral gene transfer, is the movement of genetic material between organisms other than by the transmission of DNA from parent to offsprings.
Types of horizontal gene transfer. Mechanisms of horizontal gene transfer. Horizontal gene transfer occurs through several mechanisms. Transformation. Uptake of free DNA from the environment by recipient cell. This is commonly used in laboratories to introduce new genes into bacteria. Transduction. Transfer of DNA mediated by viruses. Bacteriophages can mistakenly package bacterial DNA and transfer it to a new bacterial host. Conjugation. Direct transfer of DNA between bacterial cells through cell-to-cell contact, often involving plasmids. This is considered the most common mechanism for horizontal gene transfer among bacteria.
Antibiotic resistance spread. Horizontal gene transfer is the primary mechanism for the rapid dissemination of antibiotic resistant genes among bacteria. Genes conferring resistance can transfer between different bacterial species through transformation, transduction, and conjugation. Example, the spread of tetracycline and methicillin resistant genes in Staphylococcus aureus strains, including the emergence of methicillin resistant S. aureus, also known as MRSA. Bioremediation. Horizontal gene transfer contributes to the evolution of bacteria capable of degrading novel compounds, including pollutants and human-created pesticides. Bacteria can acquire genes for new metabolic enzymes through horizontal gene transfer, enabling them to break down and utilize these compounds.
Metabolic engineering. While traditionally, metabolic engineering utilizes recombinant DNA technology, understanding HGT mechanisms inspires new approaches to introduce metabolic pathways into organisms. HGT principles can be leveraged to engineer bacteria for industrial production of valuable compounds. Example, although not direct HGT, the concept of transferring genes into bacteria for industrial production such as insulin was inspired by the natural gene transfer capabilities of bacteria. Gene therapy. Learning from viral transduction, scientists are developing viral vectors for gene therapy. These vectors mimic the natural ability of viruses to transfer genetic material into cells. By understanding how pathogens use HGT to transfer genes into eukaryotic cells, researchers can improve non-viral gene delivery techniques.
Plant biotechnology. Agrobacterium mediated transformation. Agrobacterium tumefaciens is well known for its natural ability to transfer DNA to plants. This natural HGT mechanism is widely exploited in plant biotechnology to create genetically modified crops. Transposon mediated transfer. Transposons, jumping genes, can facilitate gene transfer between plant species that cannot cross-pollinate conventionally, offering a tool for genetic modification. Grafting-induced HGT. Grafting plants can lead to the transfer of organelles like chloroplast and even nuclear DNA between plant species, potentially creating new species or transferring beneficial traits.
Biosafety concerns. Uncontrolled HGT, especially in the context of genetically modified organisms, raises biosafety concerns about the unintended spread of modified genes. Clinical surveillance. Ongoing HGT and bacteria poses challenges for clinical surveillance and treatment of antibiotic resistance, requiring continuous monitoring and development of new drugs. Novel biotechnological applications. Further research into HGT mechanisms can unlock new biotechnological applications, particularly in areas like targeted gene delivery, bioremediation of complex pollutants, and engineering novel metabolic pathways.
Horizontal gene transfer is a powerful natural process of significant implications for biotechnology. Understanding and harnessing HGT mechanism provided valuable tools for various applications for combating antibiotic resistance to developing new therapies and improving industrial process. Continued research and ethical consideration are crucial to fully realize the potential of HGT in biotechnology while mitigating potential risks.